Hello, my name is Ryan Reeves, and today we're going to discuss immunoprecipitation and immunoblotting. We're going to look at the background information from both of these techniques, as well as some usages. We're going to look at it in a research application, and then we're going to discuss some questions that arose during the research of these two techniques here. So for some background information, immunoprecipitation is a technique that is used to extract targeted proteins or protein complexes. An antibody is used that will attach to the antigen side of a protein. Once the antibody and antigen are attached together, a insoluble substance will be added to the solution. This insoluble substance will create a precipitate at the bottom of the solution that you're testing here, and you can then extract the bottom part of the solution and have your targeted protein or protein complex. Immunoblotting is a technique used to detect proteins based on their mass, shape, and charge. A polyacrylamide gel is used because of the small pores. The further the protein migrates down the gel, usually the smaller the protein is, and vice versa. Charge is also taken into account here because of most of the solutions that are used. The negative charge is towards the top, and the positively charged ones migrate further towards the bottom. So let's discuss usages of immunoprecipitation. Immunoprecipitation is usually done to extract, like I said, certain aspects of a protein complex or certain proteins on themselves. Immunoblotting, however, is more of an identification process, though identification can be done through immunoprecipitation as well. Usually, these two techniques are paired together, and when they are paired together, you can eliminate some of the interaction with proteins or unwanted proteins. Usually gives you a little bit better result, so most of the time these are done together. So now we're going to discuss the research application portion. So as you see here, we have immunoblotting on the right hand side. This immunoblotting was pulled from a essay that looked at induction of pluripotent stem cells. The transcription factors here, as you see, were tested, and then it was found which transcription factors were best to induce a pluripotent state in a stem cell or in a cell. They tested the stem cells next to regular cells, and then they came up with the data that of the transcription factors that were best used. Strengths. This was a large cornerstone article that that did a lot for the stem cell community. So the strengths are kind of immeasurable. One of the main weaknesses of this experiment was that the results are very expensive to accomplish good results. So to push these experiments on the large scale forms would be very pricey for patients, making it very difficult for the average patient to get a hold of these induced pluripotent stem cells. But as stated, the immunoblotting was used to help identify the actual transcription factors that can induce pluripotency in these cells. So here we have a question that was formed. In immunoblotting, as stated prior, mass is a big portion of identification. And so I was wondering, if you have two proteins that are exactly the same in weight and similar in charge, what are some different methods that can be used to help identify these proteins? Or are there any other methods that can help identify these proteins? That's about all.